My name is Gilles Salles. I'm a professor of hematology in the University of Lyon, and I'm the head of the hematology department in Lyon. I'm also the chair of the CALYM Carnot Institute, which is dedicated to collaborative research in the field of lymphoma, and also chairing the cooperative group LISA. Precision medicine in lymphoma is clearly an actual challenge. We know that lymphoma is a very heterogeneous disease. We do know that we do cure many patients or have improved their life expectancy, but some patients do respond to therapy while others fail. So we do need to develop tools helping us to predict whether a given treatment will work or not, whether treatment will benefit to the patient, and also to monitor accurately the treatment of the patient. For this purpose, we have different tools which are derived from the biology or the imaging, and that we try to develop within our institute in order to develop precision medicine in lymphoma. We have learned when analyzing the clinical data from 30,000 patients accumulated over the years a lot regarding the heterogeneity of this disease. Initially, many years ago, we did consider lymphoma as aggressive lymphoma or indolent lymphoma. Now we know that any one of these entities can be subdivided based on molecular markers, molecular defect into the cells, which eventually will guide us to targeted therapy. This work obviously has been conducted by many people in the world, not only us, but our strength is really given these large cohorts of patients that we accumulate within the clinical trials, being able to translate the data from experimental work into clinical practice and bring bench to bedside. The story of development of rituximab, Rituxan, the first anti-CD20 antibodies, is very interesting because Genentech and Biogen, who were IDEC Biogen, who was the first US-based company who were property of this drug, actually aimed to develop this drug as a single agent, essentially. When we saw the early results of the drug, we pushed them to develop combination of Rituxan together with chemotherapy. And I will say that the idea of combining Rituxan with R-CHOP and assessing that in a large randomized trial in the early 2000s was actually a proposal from our group that we made to Genentech and Roche, which were linked together at that time. Later, we have been carried on a couple of studies in collaboration with Genentech and Roche, evaluating the role of rituximab either as a consolidation, what we call a maintenance treatment, but also in some indications which are rare indications, usually not necessarily examined by industry, such as primary CNS lymphoma or more recently Burkitt lymphoma, with a paper which is coming on very soon on this topic. The story of development of rituximab is a very good example of collaboration between a US-based company, although Roche is also European, and CALYM. We have now ongoing collaboration with many other US-based companies, such as Celgene, which is based here in New Jersey, with whom we have many trials and translational research programs ongoing. And as we heard during this meeting, with a smaller company, but with very promising emerging drugs such as Epizyme. If I had to give a summary of what we have learned from clinical research in lymphoma, working as a group doing both clinical and translational research, is that there is a real opportunity by collaborating early in the development of the drug to further increase the power of evaluating the efficacy of the drug, understanding the way it works, identifying better the patient for which respond to the drug, hence developing faster and more efficiently a drug by working with an academic network in this field. the ability to develop in silico models that will help us to design agents and potentially evaluate before the test in the clinic how a drug will be efficacious in a given population is just at the right of the door, at the step of the door. We are developing right now a collaboration with several companies, including a Lyon-based company called Nova Discovery, which is also here in Boston during these days, in order to work on existing databases to really identify the patient's population that will be a tool to predict in silico the efficiency of a drug on a given population. Therefore, either saying, well, we don't have to develop this drug here because this population is unlikely to benefit of it, or to select the right population where ultimately the clinical trials will be more likely to be successful. The opportunity of this FAID meeting here in Boston is a fantastic opportunity to gather with colleagues both from industry and academia here in Massachusetts, which is clearly a very well located place for research and for innovative research that will bring progress in the field. So the FAID meeting and my participation to this meeting is really aimed to further increase the collaboration between US-based colleagues, both from industry and academia, and our CALYM network.